Hi all, Dr. Clark here, and today for Gen Bio we are going to discuss population genetics. So it's kind of a continuation of the evolution topic because if you remember the definition of evolution, evolution is the change in a population's genes or genetics over time. So individuals that are population geneticists are really examining evolution or evolution in action. What I show you here is a subject we've already discussed a little bit in lecture, and that is the devil's hole pupfish. Devil's hole pupfish is probably the most endangered species of vertebrate on the planet. This is a devil's hole pupfish, and this is where they live, in this little cavern called Devil's Hole in Nevada. This is the only place they live in the world. There's no individuals coming into the population from outside. There's no individuals leaving the population. So there's no immigration or emigration. The only thing this population has is birth and death rates. So it makes for population genetics much more easy, right, to examine whether or not this population is evolving. And so that's really what this kind of section of lecture is about. How do we tell if a population's genes are changing over a given amount of time? All right, so to do this, we would look at something called the Hardy-Weinberg rule. We'll get to Hardy-Weinberg in a second here, but their research and the overall premise behind population genetics is to study the genes of a given population, whether that be fish or invertebrates or humans, whatever it might be. You're just studying the properties of those genes, often trying to figure out if the genes or the frequency of the genes are changing. And that brings us to the gene pool. And so this is what mainly population geneticists are looking at. They're trying to document all the genes or all the variations to all the genes, okay, in a given population. So if you remember back to Mendelian genetics, and I said you get one chromosome from your mother, one chromosome from your father of each of the 23 chromosomes. And on those chromosomes are genes. Okay? You have 25,000 of them. Okay? Now, dad might give you the dominant version of one of the genes, while mom gives you the recessive version, or vice versa. Okay? That difference, dominant versus recessive, is what we call alleles. It's a different variety of the gene. So population geneticists are looking at the different varieties of all the genes for a given species, okay, in a given area, okay, and over a period of time. And they're looking at how those alleles or the frequency of those alleles change. In other words, they're trying to find out if a population is fitting what we call Hardy-Weinberg equilibrium, or if it's deviating from Hardy-Weinberg equilibrium. And we'll come back to this in a little bit. Okay? So in the early 1900s, mainly once we started discovering Mendelian genetics again, so after Mendel had, published his material and someone finally eventually found it and figured out, Mendelian genetics, and we figured out what chromosomes were, and we were starting to find out what DNA was. There are a lot of biologists that question if we knew that a single characteristic is dominant, then why do we still see recessive characteristics in a population? We now know if it fits Mendelian genetics and simple Mendelian genetics, we now know that those recessive alleles hide or kind of hitch a ride on heterozygotes. So heterozygotes carry the recessive alleles, but they don't express the recessive allele. But at the time when Hardy and Weinberg were doing their research, they're interested in how does this affect a population's genetics. Okay? So in 1908, they were interested in the frequency of alleles in a gene pool. Now, I have to preface this. Remember that we didn't know what DNA was at this time. We didn't know what it looked like. We didn't know about genes. We didn't really know that there were, chromosomes that held information or anything like that. But what we did know, what Hardy and Weinberg knew, was that there were such things as phenotypes. There's characteristics, visual characteristics, that you could examine in a population, and you could see if those visual characteristics, whatever it might be, eye color, hair color, whatever it might be, you would see if those deviated from the original count. So maybe you go there to a population and you collect data in 1910, and you come back in 1915, and you see, well, is the population still the same number of dominant individuals, roughly, and the same number of recessive individuals? Hardy-Weinberg came up with kind of a general oversight or a general rule, and they did this because they sampled a lot of populations, and they found that populations that were very large and had random mating and that didn't have high mutation rates, and they didn't have influx of new individuals coming into the population through immigration, and individuals weren't leaving the population through immigration, that the frequency of those phenotypes remain constant from year to year or generation to generation. Now, remember, genotypes is the actual genetic code. We now know this, and we now know the direct connection between a genotype and a phenotype. But in 1908, Hardy and Weinberg didn't really know that, but they did know that there were physical characteristics that were inherited by offspring. And they could collect those characteristics, they could examine those characteristics, and they could see how they changed over a period of time. If they didn't change, if the frequency does not change, the population is considered to be in what we call Hardy-Weinberg equilibrium. In other words, if the frequency is not changing, then the population is not evolving. Okay? So a couple rules or a couple components to this really come around mathematically. Is it possible to determine whether or not a population's frequency of certain characteristics, phenotypes, dominant, recessive phenotypes, is that changing over time? Okay? If it is changing over time, then evolution is occurring. If it's remaining stagnant or stabilized, then then Hardy-Weinberg equilibrium is met and evolution is not occurring. Okay? So like I said before, in the early 1900s, it wasn't that Hardy and Weinberg knew what a genotype was. They didn't, but they did know what a phenotype was. And a phenotype is much more easy to determine by visual cues than a genotype is. So they could look at the frequency of a given phenotype, and because they could calculate the frequency of that phenotype, it applies to the genotype. Now this only works for characteristics that fit Mendelian inheritance. So if this is continuous variation, epistasis, incomplete dominance, codominance, the whole slew of other ways in which genetics can influence or genotypes can influence your phenotypes, then this doesn't work. But for the traits that fit Mendelian genetics, one phenotype, one genotype, then this works. Okay? And they calculated a way in which you or researchers could calculate whether or not the frequency is changing. First, they devised a very simple equation in regards to dominant traits and recessive traits. And they determined that the number of dominant traits plus the number of recessive traits has to always equal 1. Okay, especially when you're dealing with frequency. Now, if you change that to percent, so the percent dominant in a population plus the percent recessive in a population has to equal 1. Okay? And so P is denoted for the dominant individuals or dominant phenotype, and Q is denoted for the recessive phenotype. Okay, so your first equation is P plus Q is equal to 1. Okay? So simple equation, make sure that you understand that equation, but P plus Q is equal to 1. Dominant, frequency of dominant individuals plus the frequency of recessive individuals equals 1. Now, I say individuals, but this could just be a single allele that you're looking at. Brown hair versus blonde hair. Okay? Dominant versus recessive. So that allele, you could just be concentrating on a single phenotype. But often what researchers are interested in is looking at a bunch of traits. Now, you're not just concentrating on a single phenotype, but you're looking at a bunch of traits that might have changed over a given amount of time. Okay? You also have another equation. Okay? And so you had P plus Q is equal to 1, but then you also have another equilibrium equation of P squared plus 2PQ plus Q squared is equal to 1. Now, where this differs from the original equation is dominant plus recessive equal to 1. This is homozygote dominant plus heterozygote plus homozygote recessive is equal to 1. So this is just a little bit different way of looking at certain material. Now, in some cases, it's not easily distinguished, okay? Whether or not a certain phenotype is a heterozygote or homozygote dominant. But in other cases, you can. If you know how many individuals are dominant in the population, then you can calculate how many individuals should be heterozygotes and how many individuals should be homozygote recessive or homozygote dominant. Okay? So you can see this little example here. If we're looking at, 360 cats that are homozygote dominant out of 1,000, the frequency is 0.36. Okay? The number of heterozygotes is 0.48. The number of homozygote recessive is 0.16. Okay? So then if you're interested in now trying to figure out what would the frequency be if the population was 2,000, and then you can add your frequencies together, or the number of cats you have here together. Okay? So 720 plus 480, or 1,200, and divide that by 2,000. So your frequency of dominant characteristics in a population of 2,000 should be 60%. And then you can do this math of 480 plus 320, or you can just take 60% and stick it in that P plus Q equals 1 and minus the 60% or 0.6 from 1, giving you 0.4. Okay? So it's fairly easy math. It's just a simple algebra equation. Okay? But nonetheless, it allows researchers to figure out, well, if I know what the frequency of homozygote dominant, heterozygote, homozygote recessive was in year 1, and I come back 5 years later and I calculate it, have they changed? Have they significantly changed? Okay? If they have, then evolution took place. If they have not, then evolution or then that population is at Hardy-Weinberg equilibrium and evolution didn't occur. Okay, so this brings us to the assumptions of the Hardy-Weinberg equilibrium. Okay? And these assumptions have to be met in order for a population to stay at equilibrium. Okay? First, the population has to be very large. And so large enough that little minute changes here and there are not going to affect the population. Okay? For example, the human population is very large, 7.7 billion people. Okay, that's a very large population, and it's highly unlikely that a single mating is going to make a single bit of difference in the population. Individuals have to be able to mate at random. Okay? And so there is no mate choice. Right? Organisms aren't picking organisms based on color of the feathers, their ability to sing, how tall they are, what's their muscle mass, how much money they have in their pocket, anything like that. There's no mate choice. Okay? It's purely random. Okay? There's no mutation. Okay? So the population itself is not, the genes are not mutating. You're not making mistakes when copying DNA. There's not a high level of mutagens. There's no mutations. No immigration or emigration. So the populations are stagnant. There's nothing coming in and nothing leaving. Okay. And natural selection is not occurring. Okay. So in other words, nature is not selecting for one type of allele versus another type of allele. So from generation to generation, the probability that one allele will survive versus another is the exact same. Natural selection is not occurring. So you might ask yourself, well, that seems crazy. It doesn't seem like there's really any population of any organism on the planet that would fit those 5 assumptions. Some human populations potentially could fit that bill, though. Okay? Population is very large. Most of the time, mating is purely random. You don't know that other individuals' characteristics. There's very few humans that say, oh, I'm going to mate with that individual because they have blue eyes or blonde hair or because they're 6 foot. Okay? So most traits are purely random when it comes to mating. The one thing that doesn't occur here that human populations don't fit Hardy-Weinberg equilibrium, and that's because we have no control over mutation rate. We have no control over immigration and emigration typically, and natural selection can take place, and it can be part of human populations. But human populations are probably the closest group of organisms that actually fit Hardy-Weinberg equilibrium, and we don't fit it very well. So why was this ever brought up? Okay. Well, in part to show people that most populations are not stagnant. Most populations are violating Hardy-Weinberg equilibrium, and they're actually evolving. And the other piece of this, though, really came from Hardy-Weinberg equilibrium, is to suggest why we still have recessive genetic disorders in human populations. So you have sickle cell, you have cystic fibrosis, you have these recessive genetic disorders that still exist in the human population. And part of that reason is because we have complete random mating, okay? And the population is really large, so you still have these recessive traits that are hidden in the heterozygotes and just keep being carried on and on and on, and there's really no cost to having those recessive genetic disorders in the population. So that brings me to the 5 factors that will deviate or change the amount of homozygote individuals and heterozygotes in a population. And you should be able to kind of figure these out based on the characteristics that will allow homozygotes and heterozygotes to stay the exact same over a period of time. You can pretty much put the opposite out there. If you have high mutation rates, if your mating is non-random, so there's mate selection. If you have genetic drift, we'll get to genetic drift, but that happens in really small populations, and it's like flipping a coin 10 times. You know that flipping a coin 10 times, a fair coin, you should get 5 heads and 5 tails. But rarely are you actually going to get that. Okay? You just, by chance, might get more tails than you do heads. This is the premise of genetic drift. By chance, you might get more recessive traits than dominant traits. Okay? And so you can get this swap of heterozygotes, homozygotes, okay? And you can get this pendulum shift, okay, from dominant to recessive very quickly, especially in very small populations. Migration of individuals out of a population or into a population can bring new alleles or remove alleles from the population. And then, of course, any type of selection. Natural selection, sexual selection, artificial selection, any of these can cause a deviation of the normal allele frequency of a population. So let's look at these. Mutations, we've already talked about them. Remember, this is just a nucleotide change. So it could be at a single point. It could be a point mutation. It could be a chromosomal mutation where you have translocation of a chunk of chromosome. But a mutation is just a change in the DNA sequence. Now, as long as you don't have a huge amount of mutagens in a certain region, mutations alone are not going to drive evolution. They just don't happen often enough to change enough genes that the frequency of alleles could change in a population. But what they can do is they can be agents of evolution. In other words, if you have a new allele or a mutation pop up, okay, natural selection or sexual selection can act upon that mutation. Okay? So alone they might not drive evolution, but coupled with natural selection, they're huge drivers of evolution. Okay? The other piece of the mutation that makes it interesting is that if a mutation happens in a somatic cell, okay, it's never passed on. So the mutation has to be passed on, or has to happen in a germline cell to be passed on to the next generation. If it's in somatic cell, it's not passed on, and it really makes no difference. Maybe for the individual, it makes a difference, but not for the population's genetics. Okay? And like I said before, no matter how often they happen or how rare they are, in the end, mutations are what gives us those new alleles. It gives us that new variety of a gene. And by doing so, then other agents of evolution can act upon it. Natural selection, sexual selection, even artificial selection. Okay, non-random mating. This is pretty self-explanatory. This is pretty easy to understand. Okay, this is just when mates are chosen based on some type of phenotype. Okay, and you're going to get certain genotypes pop up and be more common than other genotypes. Okay. Sexual selection, Charles Darwin came up with sexual selection, and that's one excellent way of demonstrating non-random mating, is that mates are chosen based on some physical characteristics. There's another way at which you can get non-random mating. It could just be that organisms have to go to a given region to mate, and only the organisms that are fit enough or physically capable of making that journey would then be able to mate. Okay? And so that's another type of selection. Individuals that are weak are not going to make the journey, they're not going to mate, and ultimately their genes will not be passed on to the next generation. Genetic drift, okay, like I said before, this is just random change, okay, and it's just a result of really small populations, and you can get big shifts in the allele frequency. This is what's probably occurring with a lot of endangered species, is their populations get so small that you can get fixation of things like recessive alleles, or even dominant alleles can be fixed and there's no variation in the population. Everyone has that one gene and it's fixed, so there's no variety, which can cause a lot of situations when, say, disease outbreaks come through, or a new food type is introduced, a new predator is introduced, a new prey is introduced, a new competitor is introduced. It can cause huge issues for that organism. Okay, so like I said before, it occurs in small populations and can result in harmful effects in the sense that you can get a bunch of alleles that are fixed, and so there's no variation, and this can ultimately reduce physical output, so reproductive output, or even cause death in a large portion of the population. Okay. We'll come back and we'll talk about genetic drift when we start talking about like population bottlenecks and founder effects and things like that. Okay. Migration. So migration is movement of individuals between populations. This can cause a population which has been stable and had a stable allele frequency to deviate from that because you have an influx of new individuals or an outflux of some of the phenotypes. And it's really just based on a couple things. First of all, how many individuals are migrating? So what's the new influx or the outflux going to be? Okay. And then how different are the alleles of the individuals that are coming into the population or leaving the population? How different are those alleles? Are they enough to change the frequency of homozygote dominant to heterozygote to homozygote recessive? So when we talk about these agents of evolution, we need to talk about them collectively. And that rarely do these agents of evolution occur by themselves. So yes, we talk mutation. You're going to have mutations in your DNA just through, poor copying of your DNA as you're developing. Well, some of those mutations, if they're happening in germline cells, they could be passed to offspring. But in order for them to be passed to offspring, the individual still has to be able to reproduce. And in some cases, the individual has to have a characteristic which a mate would choose them. Right? So you might have mate choice and mutation. Or you might have natural selection, which removes individuals that have certain characteristics and allows other individuals to survive. Okay? On top of it, if you have mutations pop up in a population, and along with that the population is very small, you can get genetic drift to occur, where you're fixing some of these mutations, whether they be maladaptive, so they might cause harm to the individual. You're fixing them into the population because the population is so small. And so there are a couple ways at which this occurs fairly often. Okay? One where it's not the influence, or not really influenced too terribly much by humans, except for when we move species from one area to the next. Okay? But a lot of organisms will migrate to new regions, okay, regardless of the human effect. And that's called the founder effect. When you have a few individuals, so you have a set amount of genetic diversity, when those few individuals move to a new region, and get established, that new population is just based off of those few individuals. And you can get some situations where you get fixation of some genes that are, very specific to that population. The other piece that humans directly have a hand in it, and do this on a day-to-day basis, is bottleneck species. So the bottleneck effect is when you have large population with lots of genetic diversity and it shrank down to a very small population. That shrinking process, you lose a lot of your genetic diversity. This happens when species go from, very abundant to very few, and then even if we step in, we try to reestablish that species and bring them back, the genetic diversity is so little that the individuals really don't stand a chance. So, things like bison, grizzly bears, mountain lions, devil's hole pupfish, cutthroat trout. There's just all kinds of species that have gone through genetic bottleneck effects, where the populations used to be very big, and now they've been shrank to a very small size. And then after we realize that they're on the brink of extinction, the bald eagle, right? The brink of extinction, then we bring the population back. Right? But the genetic diversity is already very low. Okay? So here you can see genetic drift or bottleneck effect. So you have a big population, a few individuals make it, so the population is drastically cut, maybe through hunting, maybe through fishing, maybe through habitat destruction, all kinds of things. The use of DDT, all kinds of things can cause the population to shrink. Okay? So you got very few individuals. Then we step in, we give them habitat, we ban DDT, we stop the hunting and stop the fishing, and those surviving individuals rebound, and the populations get back to what they used to be, but now you're missing some of those genetic alleles. You're missing that diversity, okay? And that's part of genetic drift. Okay, so selection, we've talked about it before. Darwin was, kind of the developer of the mechanisms for evolution from the standpoint of selection, and was really kind of this, the driver of, discovering how either nature can select for certain characteristics, or humans can select for certain characteristics, or different sexes can select for certain characteristics, and that's really kind of the general premise behind Charles Darwin's work. So artificial selection, like I've said before, Charles Darwin was well versed in artificial selection. He raised horses and cattle and pigeons and things like that, and he knew lots of individuals that also did this, and he got lots of characteristics, and a lot of the evidence he had for the ability for species to change really came through artificial selection. Natural selection, or this ability for nature to determine what organism is going to survive, and what characteristics are most suited for that current environment. And this is pretty simple. Most of you understand natural selection fairly well. Is that you might have dark-coated mice here, and you might have two coats. So these dark-coated mice have the capability of producing a light-coated mouse. Okay? So maybe recessive is light and dark is dominant. So they have the capability. But the reason why we don't see these light-colored mice on black rocks is because predators see them before us. So the predators will take them out of the population, and only the dark-colored individuals are there. You move off the basalt fields, off the dark-colored rocks, and out into the sands, and now you have the opposite effect. Okay? Light-colored mice, yes, they might give rise to a dark-colored mouse, but it doesn't last very long because the predators pick it off. And so you get a bunch of light-colored mice in this region that match their background. Okay? And that's natural selection. But natural selection occurs in three main types. Okay? You can have what we call stabilizing selection, where the extremes, okay, maybe in this case it's showing body size, the extremes are selected against. Okay? So you have this truncation of organisms into, this realm, this normal realm. Okay, and you can think of that from lots of standpoints. Lots of human characteristics fit a stabilizing selection. Okay, height. There's no 10-foot individuals and there's no 1-foot individuals. Okay, so we're stabilizing, body size in that. Okay. Disruptive selection. That's where the middle portion, the individuals that might be intermediates, are selected against. Okay? And a lot of like beak design, so you either have a small beak or a large beak, but a medium beak, you're at a disadvantage. Other things like even body size. You might be small or you might be large. Small individuals are what we call sneakers, and they can sneak in and reproduce. Large individuals might hold territories and stop any intermediates, any medium individuals from ever reproducing. So eventually over time, you might have two separate species because either you're small or large, but nothing in between. And then directional selection is going to be selecting for one of the extremes. Everyone's bigger or everyone's smaller. And so you get these big peak shifts that move in one direction. And we'll look at some other examples. Okay. So stabilizing selection, or selecting against both extremes. Okay. So the extreme phenotypes are basically eliminated from the population. There's lots of examples. I said height, things like that. Probably the best example is human birth weight. If you think about the average birth weight of a human, it sits at about 7.5 pounds. Okay, or 7 pounds is the average weight of a human. Okay, and this is pretty much stabilizing selection. Now, that being said, you do have situations where modern medicine and modern technology has kind of stretched this stabilizing selection out. If you think about babies that were born in the 1800s or the 1700s, okay, if they were sitting at 2 pounds or 3 pounds, there's no way they're going to develop. Okay? Preemies didn't really have a chance. It just wasn't going to happen. But the same thing occurs on the other end. If a baby was going to be produced that was 10 pounds or 9.5 pounds, and the mother had to have a C-section, in the 1700s, that really didn't occur. So the mother would die and the baby would die, and those genes would be eliminated from the population. Thus, the population's weight of children was somewhere around that 7-pound mark. Okay? And so that's a pretty good example of stabilizing selection. Now we've definitely stretched this out because now babies of 2 pounds and even less than 2 pounds can make it due to our incubation and modern medicine. And same thing, babies over 10 pounds can make it with C-sections and things like that. All right, disruptive selection. So you're selecting for one of the two extremes or the two extremes and nothing in between. So there's no middle ground. And like I said, this often happens with kind of the bills of birds. And this happens with the Galapagos finches in many cases. But we also have good examples of African black-bellied seed crackers, and they're going through the same sample. They're going through the same example. Either you have a big bill and can crush big seeds, or a small bill and can handle very fine seeds. But in between, if you have a medium-sized bill, you're going to be outcompeted by the big-billed individuals and outcompeted by the small-billed individuals. And so that's a fairly good example. Now, often when you have disruptive selection, that's going to eventually amount to speciation. You're going to have two separate species. Directional selection, okay, so this is selection of one of the extremes, okay, and there's tons and tons of examples. One of the best examples is experiments that have been done in the lab, where you can take things like fruit flies, and fruit flies naturally will fly towards the light. And if you place a light over, say, a fruit fly container, the fruit flies that fly towards the light will die. Okay, they'll get burned up and they'll die. And the fruit flies that don't fly to the light or avoid the light will tend to reproduce. And so over a very short period of time, you can get kind of the average number of individuals flying towards the light to drastically decrease. So you're shifting your population, individuals flying towards the light, to individuals that don't fly towards the light. Very easily done. Okay, 20 generations. This can be done in 3 months or so, maybe 5 months max, and you can get this drastic shift of a population. Okay, so those are some examples of natural selection. There will be lots more to come as we progress with evolution. The next topic we're going to talk about is evolution in action. So I'm going to give you some examples of studies that have shown evolution occurring within a given amount of time, a relatively short period of time, and there's some really good examples of how evolution or how populations can change over a small period of time.